Hi, I'm Hannes Stärk, a PhD student at MIT working with Regina Basilei and Tom Yakula. And I'm excited to be here today on the behalf of this team to be presenting BoltzGen, our new binder design model. And to motivate the need for such a model, I want to start with an example. Here in blue, we see third telomerase, a target or a protein that our collaborators have identified as an important target against glioblastoma. And what this protein does in glioblastoma cancer cells is to repair DNA. And now we want to stop this DNA repair and then stop the cancer cells from surviving by designing a peptide that binds at this location and displaces the DNA. Now, to do this, the existing frontier models would already fall short in just a single drug discovery example. First, they do not support the required constraints that the human expert want to impose here on the design. And second, third telomerase is a novel target in the sense that there are no existing bound proteins or peptides to this target. And the existing frontier models have not been demonstrated to generalize to such novel targets. So what we want to do with BoltzGen is to expand the designable space of binders once along this axis of generalizing to modalities and constraints that the human experts want to impose. So we want to have one model to design nanobodies, peptides, proteins, and to target proteins, small molecules, DNA, RNA. And second, maybe even more importantly, we want to now be able to generalize to novel targets, right? I said that the previous models, they have virtually always been evaluated on targets that are already in the training data with existing bound structures. Now we want to get away from this and also be able to generate binders against novel targets in this sense. And to do this, we think we require getting closer toward emulating physics and a model that is strong at reasoning about structure. But before I now talk about how we achieve such a model, I want to interrupt by saying that, of course, to test whether we achieve these two axes, we need to bring the designs into the physical real world and test them out in the wet lab. We do this in collaboration with now more than over 30 collaborators, and I'm happy that 8 of these wet lab campaigns have already finished, and I'll be later able to show you the results across 26 targets. And now back to the model. What we want to achieve is, on the one hand, strong structure reasoning, on the second hand, emulating physics, as I laid out on the previous slide, and then we want to have this notion of full controllability. And now, to explain each of these points, I will have one slide for each. To make very explicit why we think strong structure reasoning is required, then let us consider what does it mean to design. Well, we have our target A, and now we want to find a binder. Finding the binder means discriminating between the infinitely many possibilities of what the binder could be, like B, C, D, and so on. And discriminating between B, C, D being the binder means you need to understand how A would interact with B, how A would interact with C, and so on. And understanding how biomolecules interact with each other, that is exactly the folding task. And maybe in other words, if we want to do strong structure-based drug design, strong structure-based binder design, then we need to be strong at reasoning about structure. Point number two, we want to get closer toward emulating physics. With this, I want to say that even if we were only interested in designing nanobodies that bind to proteins, then we would still want to train our model on proteins binding to proteins, for example, because the context, the patterns that matter, they appear in all these types of data. And if we want our model to now pay attention to use patterns that generalize well, then these are the patterns that are present in all modalities, like, for example, physical interactions. So we want to exercise our model in as many contexts on as many modalities as possible, such that the remaining patterns that are not spurious are well generalizing patterns such as physical interactions. And point number three, what do I mean with controllability? Here, just based on an example, well, sometimes our collaborators want us to design only the CDR3 region of a nanobody and then keep everything else, including its structure and the sequence, fixed. And sometimes they want us to redesign all CDR regions while keeping the scaffold fixed. Sometimes we want to redesign everything. And now this is the spectrum of possibilities along one axis of controllability, so to say, this partial design aspect. And we have the spectrum across many other controllability options. But these are now the 3 points. So how do we achieve them? Towards strong structure reasoning, we turn, so to say, the design task into a structure prediction task. Toward emulating physics, we exercise our model on as many contexts as possible on the available data. And toward full controllability, we have an expressive design specification language that lets users specify essentially any constraint that they might want to have on the resulting binder. And now, before I go into details of these model aspects, I want to say that the model that I'm talking about right now, this is always only the first part in the larger BoltzGen pipeline. It's maybe the most important part, but the other parts are also crucial for getting out good final designs. But now, how does this first part work? What is happening here is that, oh, well, what we're looking at first is one generation trajectory, one denoising trajectory to end up with one binder in yellow here against this blue target. And what the model is doing is it's simultaneously folding the blue target, then predicting the conformational change of the blue target while binding the binder that is simultaneously being generated. And then we're generating the binder. What does it mean to generate the yellow binder here? Well, we're generating its 3D structure and its sequence. So we have this joint continuous and discrete generation problem going on, joint structure and sequence generation. And how this is solved in our model is that we start from complete noise for both the target protein and for the design protein, and then only produce 3D coordinates. The only output of the model is a 3D point cloud. And the sequence of the target is just fed in as additional conditioning information to the diffusion model. And this is quite distinct from how existing models have usually tackled the problem, where usually the target and the design protein, they would be treated very differently from each other in that for the target, the denoising, the diffusion model would already take the clean structure as input, which means it learns to just copy that structure over and no folding is happening. The model is not learning these expressive structure prediction features that then will also help us for doing strong structure-based design and having strong structure reasoning. In our architecture and our approach, this is arguably much simpler and more scalable, and we only produce this 3D structure, right? So this comes with the advantage if we now have our noisy target as well, that we're repredicting, that we can reason about the conformational change, which is important when, for example, binding to intrinsically disordered targets. But now this joint generation of sequence and structure that is still happening here. But the sequence generation, the discrete part, is embedded in the continuous generation part. The geometry that we're generating directly encodes the residue types that we're generating. And how this is encoded, we see on this slide. Because for us, every designed residue now has 14 atoms. So even if you, for example, generate an isoleucine, which has 8 real atoms, right? Then for us, this isoleucine now instead has 14 atoms, where we have an additional 6 virtual atoms that are placed at the same identical position as the oxygen of this isoleucine. So during training, the model is tasked to produce this point cloud of 14 atoms if it is tasked to produce an isoleucine. And with this, we have now turned the design task into a structure prediction task, right? Where now for designing, we're also just producing a 3D structure. And point number 2, toward emulating physics. What do I mean with exercising a model on as many contexts as possible across as many modalities as possible? Well, we will just see this based on an example. What we do during training is we sample our PDB structure or our structure from PDB and then have this large complex here. And then we randomly, right, again, random because we want to exercise our model in as many contexts as possible on this data, sample a crop center, which is just a residue of this complex, like, for example, this residue here. And then we end up with this subcrop that can be larger or bigger of the protein. And then on this crop, we exercise the model to, for example, only predict structure, meaning that the model gets as input the sequences of both chains here, and then just is tasked to predict the 3D structure. Sometimes we train it with the design task, where now for the yellow region, the model no longer gets a sequence as input, and it is tasked to produce this 14 atom representation. Sometimes we feed the structure of the target or a partial structure of the target as input. Sometimes we feed the binding site as input, and so on and so forth with all the other control options that we will see soon. And also, this task distribution changes over the course of training, where at the beginning of training, for example, we only train with structure prediction. And that's it for point number 2. Now I want to lay out what are all these control options that I mentioned when I mean that we want to allow for full controllability. Well, first, this partial design aspect we've already seen. You could say, for example, you want this to be a cysteine, this to be a cysteine, and all the other residues to be designed. And then you can say that you want the secondary structure of this thing, for example, to be a helix. You can say that you want a covalent bond between the cysteine and the atom of another chain, or you can specify disulfide bond or any other covalent bond you might want. Then you can specify not only the site where you want your design to bind to your target, but also which are residues in your target that you do not want to bind to. And now, lastly, to the structure control that we provide here based on an example where we want to design a nanobody here in orange and yellow to bind to this blue target. And what we want to say is that the structure of the dark orange scaffold should be fixed. The structure of the blue target should be fixed. But the structure of the target with respect to the nanobody should not be fixed. The nanobody location and orientation with respect to the target should be left up to the model and decided by the model how it binds and where it binds. For this, we have this concept of structure groups and visibilities, where if something is in the same structure group, then its structure with respect to each other is specified. But if it's in different structure groups, then they are free to move with respect to each other, but still have their structure internally specified. And that's it. All these control options are fed into a sort of design specification language where you can go ahead and write a file that is very expressive and simple to specify, and then the designs that come out will adhere to the constraints that you desire. And with this, we now have our design model, this generative model built up, and can get back to how this fits into this larger pipeline. Well, what we would do is we have our design specification language, put this into our generative model at the beginning, then we generate, for example, 60,000 designs. Then we run an inverse folding model where we refine the sequence. And then we run a refolding step where we extract the sequence, the molecular information from the design structure. And then we run a folding model to repredict the structure. And then we hope, we check, is the repredicted structure the same as our generated structure, our design structure? And then we use this information later in the filtering. We have another affinity prediction step that I'm skipping now. And then we have an analyze step where we check physical metrics like the number of salt bridges, the number of hydrogen bonds, the surface area that makes up the interaction between the binder and the target, and many more. And all these metrics are then fed into a final filtering step where we combine the metrics in a different weighting and then come up with a final ranking, and then run a diversity quality optimization algorithm that chooses a final set of experiment budget, many ranked diverse designs from the set. And now there's a little break. We are looking at some denoising trajectories here before we look at a few results. And the first set of results that I want to look at is computational before we get into all of the experimental wet lab results. And the computational results that I'm looking at here are just structure prediction results. So there's no design going on here at all, right? And the reason we're looking at this is because we argue that to do strong structure-based design, you need to be strong at reasoning about structure. What does it mean to be strong at reasoning about structure? To be strong at structure prediction. And we indeed find that here with BoltzGen, we have a model that can design new binders, and it can also do structure prediction. And its structure prediction performance, in fact, reaches the state of the art of structure prediction models. so what we see here in blue is the BoltzGen structure prediction performance, and in yellow we compare this with Boltz-2. And this also holds across modalities, not just for the interprotein metric. But now to the wet lab results, right? I said this, we have these over 30 wet lab collaborators, and then we now have the results from 8 wet lab campaigns across 26 targets. And before I dive into some interesting case examples of our wet lab collaborators, I want to look at a systematic evaluation of generalizing to novel targets in the sense of targets that do not have anything in the training data that is similar, anything in the training data with a bound structure that is similar to the targets that we're considering now. And right, remember, this is very different from how existing binder design models have been evaluated, where the targets usually are in the training data with existing bound structures. Here we now select 9 targets that have less than 30% sequence similarity to any bound protein in PDB, so they might not even have suitable binding sites for example, a nanobody. And then for each of those, we design 60,000 proteins and then test 15 in the wet lab, check how many express, how many are binders, and what is the best binding affinity. And we find that for 6 out of the 9 targets, we're able to get nanomolar binders with just testing 15 designs per target. And also, all these designs have no off-target binding against human serum albumin, a very interactive and abundant off-target. And the same results also hold across mini protein design. Now to one example here where we collaborated on designing a binder against this green intrinsically disordered region of this protein here. And then we designed peptides against them. Our collaborators tested 5 of them in the wet lab by attaching a fluorescent tag to the peptide and then putting it into a live cell and then checking where does the fluorescence in the live cell now occur. And then we find that it only occurs in the nucleoli of the cell where we know that the target is localized, which then tells us likely it binds to this target. And second, it also tells us that likely it doesn't bind to anything else in the cell that is not also localized in the nucleoli. Then another example here, we try to design proteins to bind 3 different peptides that are antimicrobial or hemolytic. Hemolytic meaning rupturing red blood cells. And also interesting about these peptides is that they're very differently and diversely structured. Some are even disordered. And against these, we're now always able to obtain at least one design when only testing 6 in the wet lab that neutralizes the biological activity of those peptides. Also, for 2 of the targets, we again get nanomolar binders when just testing 6 and considering the entropic penalty that you will incur when binding such a disordered peptide. And lastly, we're looking at the fact that we're also succeeded at designing some proteins to bind small molecules. Here, one target where again, we just tested 6 designs, obtain our binders, and we now have also other binders against prelacidin, for example, where we also only test 6 and obtain nanomolar binders. With that, I want to conclude that we set out to expand the designable space across modalities and constraints and to novel targets in our sense, right? And to try to achieve this, we turned the design task into a folding task. So we unified the two. Then we exercise our model on as many contexts on the underlying data as possible in an aim to get the model closer toward emulating physics and learning physics based on example. And 3, we have this design specification language that is very expressive and lets the user specify essentially any human constraint that one might want to have on the designs. And then lastly, we tested this now on the results of more than 8 wet lab campaigns across 26 targets. And with this, I want to say that maybe we now have a model that is fine at designing binders, de novo, but we should keep in mind that affinity is only the first step toward, for example, designing a therapeutic, where we would also have to care about delivery, degradation, immunogenicity, and similar. And of course, we are working on these things. And with this, I want to conclude and finish with a last thank you to our collaborators that took a chance on our designs and tested them in the wet lab. Here are the links to the paper, to our GitHub, our Slack community, and we would be very happy to collaborate with you if you're interested. So feel free to reach out and have a great day.